met in Makerere during the first East African Phage Forum. By then, we had initiated to do some phages, but we did not have proper skills. I want to say that Dr. Tobi and the team, that is Professor Mother, Clocky, Dr. Janet Nall, and Dr. Ben Chan, they really initiated us proper into the phages. They imparted the skills onto us. Tobi leads Phages for Global Health, which is a charity organization that facilitates the application of phages in Africa and Asia. And as we know, 90% of the deaths from antimicrobial resistance are expected to occur in Africa. Tobi previously spent 15 years working in the pharmaceutical industry, co-developing drugs that have been tested in more than 80 clinical trials worldwide. So for those who are thinking about commercialization, I can tell you we have the right person in the forum. Dr. Tobi holds a PhD in medical engineering from Harvard MIT Division of Health Sciences and Technology. You know, when we are MIT or Harvard, then you know that we really have a top-notch speaker today in our forum. So, and I want to welcome her to present. And meanwhile, I want to recognize also the presence of many of our collaborators, including Professor Skanik, who has been very instrumental in the capacity building. He's the one who gave a PhD to Dr. Michael Chiang, who was my PhD student. So Dr. Tobi, it's your time today. We are happy to have you around, and we thank you for having found time to be with us. And we also appreciate for your time because you have spent a lot of time trying to train Kenyans, Tanzanians, and Ugandans. And I know you went to West Africa. You are doing a lot for us. Thank you very much and welcome.
Thank you very much. It's nice to be among friends again virtually, and I really do wish I could come to Africa sometime soon. Let's pray for the end of the pandemic. So I do want to thank you for that introduction. I don't think I need to say anything else except to jump right into the talk. And I want to start by comparing pandemics that we have endured as a global community, starting with the AIDS pandemic. That killed roughly, if we look at the millions of people killed worldwide each year, that pandemic killed roughly 2 million people at the peak, at the worst year, which was 2005. And as I'm sure you know, roughly 80% of those deaths occurred in Africa. Then we're, of course, in COVID, where also in 2020, roughly 2 million people were killed by that virus. And we're almost done with 2021, and nearly 3 million people have been killed so far worldwide by COVID-19. But as I'm sure you know, the antimicrobial resistance, or AMR crisis, will be even bigger. 10 million people expected to be killed by 2050. And there's three parts of this that make it particularly challenging, I think. Not only the vast number of deaths that will be caused by AMR, but if you look at comparing those to AIDS and COVID, each of those were basically caused by one virus and variants of those viruses, as we're seeing, but really one virus each, where AMR will be caused by many different bacteria. And then, as Atunga already says, and as I'm sure you're familiar, this is a image adapted from the O'Neill AMR report from 2016. 90% of the deaths from AMR will occur in Africa and Asia, which is why Phages for Global Health focuses in those regions, where less than 400,000 deaths will occur in Europe, the US, and South America, and even fewer down under. More than 4 million deaths each will occur in Africa and Asia each year. So I want to look at how we've developed and delivered drugs, we being a global community, for each of those pandemics, and what we can learn from that and hopefully do differently for AMR. So starting with antiretroviral drugs and AIDS-related deaths, this is a figure that was published by John Nagel, the leader of the Africa CDC, last year in Nature, and looks at the total number of deaths in Africa over these years as compared to the number of deaths in the US, which is much smaller. And in particular, it looks at when antiretroviral drugs were delivered. This little blip right here in blue is when antiretroviral drugs were first introduced broadly in the US. And you can see that the deaths went down significantly, even though it's just a tiny blip. But it was over 10 years until antiretrovirals were really available in Africa. And during that time, there was all these millions of deaths occurring. And then once the drugs were available, the death number started to go down somewhat. And when Nagel published this paper, his intention, he did it near the beginning of the COVID pandemic, and his intention was to say, please, let's not do that again, essentially, for the COVID crisis. And unfortunately, I think we're seeing the same problem, as you know, with the vaccine distribution globally. So if we look at coronavirus vaccination rates, and here I've taken 6 representative countries from Europe and the US and 6 from East and West Africa. And this is data I downloaded from Our World in Data about 3-4 weeks ago. So the numbers have changed a little, but not much, unfortunately. And the numbers show in dark green the number of percentage of the population that has been fully vaccinated against COVID, and light green is those who have been partially vaccinated. So it varies between 68% and 82% in US and Europe, as opposed to 2.4% and 9.9% in these African countries. And I despair when I look at these numbers, because we need to do better as a global community in addressing these pandemics, especially when it comes to the bigger one, AMR, that's coming. And so let's think about how has drug development and delivery been done, and what can we do differently? So the overall simplified model is that drugs are developed in industrialized countries, and then those are distributed to developing countries. But the big problem is this delay that I've just shown data for. And of course, there are many reasons for that delay. There's financial support that needs to come from international sources usually, as well as the supply, and there are storage logistics. We know with some of the COVID vaccines, they required -80 freezers, which aren't always available in some parts of Africa and Asia. And then was some hesitancy, and I think there still is, in cultural uptake of a drug that's developed overseas. And so to overcome this, is it possible for developing countries to make drugs for their own populations? That's the goal I'd like to see us achieve. And let's acknowledge that it's particularly complicated for AMR because multiple drugs will be needed for the different bacteria, not just one virus like COVID. And if we were talking about traditional, conventional drugs, small molecule or antibody-based drugs, this would be an even more challenging task. But since we're talking about phages, I do think we have the possibility, anyway, of achieving that goal of making drugs in country. As you know, we're all here because phages are well suited for developing countries. They can be isolated in country with equipment that is locally available. In fact, it may be best to do this in order to address local bacterial strains. It's potentially less expensive to develop phages than conventional drugs. And this could be a whole nother talk in and of itself, that the antibacterial drug market is not economically sustainable. Oops. And let me just say that companies that have successfully made new antibiotic drugs have gone bankrupt in the past few years, 2 or 3 years, because they can't sell those drugs for enough to cover the cost of making the drugs. And so this is a worldwide problem. And so especially for the AMR drug market, we really need to keep the costs low. And phages are better suited for that, I believe, than conventional drugs are. Local production of phages could also overcome several of the issues I highlighted in the previous slide. There would be fewer logistics in delivering them to these countries. There would be in-country control, so less dependency on industrialized countries. Hopefully, there would be improved cultural acceptance of the drugs if they were delivered in a quality control way. And phage banks could provide tools for killing different types of bacteria, since we're going to be dealing with a broad spectrum, unfortunately, of bacteria in the AMR crisis. So let's talk about how phage banks in particular could address this. And we know that phage banks started in the former Soviet Union, and the two that are most famous, if you will, are the Eliava Institute in Georgia and the Herzfeld Institute in Poland, where thousands of patients have been treated successfully, I believe, with phages. But it's fair to say that a number of other phage banks now exist around the world or are in the process of being established. In the US, in both the US Navy Medical Research Center and the Walter Reed Army Institute of Research, they have established large phage banks. The Queen Astrid Military Hospital in Belgium also has a phage bank. Adaptive Phage Therapeutics has actually set up a company based on the phage bank idea, where they have large collections of phages. And a number of phage therapy centers that are based in academic centers are now popping up in the US, several in the US, in Israel, in Australia, and in the Netherlands. So this is not just a concept, the phage bank concept that exists in the former Soviet Union anymore. And so what if we were to establish phage banks in Africa? What would that look like? Well, we're envisioning that these would be national banks managed by centralized authorities. They would be hosted at government-funded research institutions or universities. And phages could be contributed to those collections by scientists from throughout each country. So that could really add to the strength and breadth of the phages there. We're also envisioning, I think realistically, that the phage banks would need to be supported by international public and philanthropic funds, although, which is the same as drug development at the moment, drugs being developed and then delivered to developing countries. But I think it would be more cost effectively to do these phage banks in country. Undoubtedly, public-private partnerships would be needed to implement, meaning manufacturing, distribution, purification of the phages at scale. And international phage expertise and collaborations are also needed, at least initially, as the capacity building continues in Africa. Our thinking has been that it would be best to develop phage cocktails that target bacteria that are found to be circulating in the general population. This would be different than an individualized phage therapy approach, which is being done in some places in Europe and the US. And ultimately, I think to make it work, these cocktails would need to be provided at cost or at supplemented rates. In other words, it would be for public benefit, not for profit. So not through commercialized venues, since it's very difficult, frankly, to make it successful and sustainable if this is a for-profit venture. So in a nutshell, those are the thoughts that several of us have had about establishing phage banks in Africa. This is no small undertaking, and I would love to hear if there's any thoughts or differences in opinion too about how this might be done. But I think this large undertaking needs to happen if we're going to address this huge number of deaths that will take place in Africa. And let's talk about what would be needed to establish phage banks. This isn't just a refrigerator or a freezer or two in a laboratory. This needs to be a broad, standardized management system with following general guidelines. So for example, accreditations such as ISO-CAP or MIABIS, which stands for Minimal Information About Biobank Information Sharing, and ISBER is International Society for Biological and Environmental Repositories. These are some international standards that are out there. Those types of standards would need to be applied for sort of quality controlled phage banks. This would include monitored storage facilities with access controls, backup power, stable and secure databases, really rigorously controlled systems. The same would be true for any protocols of how the phages were characterized, produced, purified, stability testing of those phages, routine quality control of those phages. This would need to be a machine that's just a regularly set up, by a machine, I mean a system that is running in a very controlled way. That requires the bottom bullet point there, technically competent staff, and hopefully there would also be host strains that available that could be integrated with local AMR surveillance programs so that you could be regularly testing those phage collections against AMR surveillance program bacteria that had been identified. And of course, there would need to be networks within countries for distribution of the phages. Maybe you could use the same networks that have been used for antiretroviral drug distribution. And it goes without question that there would need to be oversight from regulatory authorities who need to clarify how they want to oversee phages, IRB committees and ethics committees. So again, this is not a small undertaking to establish these types of phage banks. Now, let me say that I have been talking with the groups around the world in Israel, Australia, Germany, Belgium, who have established national phage banks. And we're talking about initiating an international working group that would include collaborators from those existing phage banks and those such as from Africa wishing to start new phage banks. The idea would be that this group would draft guidelines for these management systems, characterization of the phages, manufacturing, purification, all of the issues I just spoke about. And through that group, we could also identify experts who could be available for consultation on how to set up, manage, and implement phage banks in Africa. So we're working on getting such an international working group going and hopefully getting some funding to pull formalized meetings together to establish this. The other thing that would need to happen, excuse me, is that we would need to get full characterization of the existing phages and phages that will be continuing to be isolated in Africa. I know that the Africa Phage Forum has already done a survey of what phage work people are doing there. We also have a database of phages that have been isolated from people who have gone through the Phages for Global Health training workshops. What we'd also like to do is identify what else is needed for those phages in terms of, say, DNA sequencing, expanded host range analysis, TEM analysis. And ultimately, Phages for Global Health would like to help connect scientists in Africa who have been isolating phages and beginning to characterize them, connect you with potential collaborators within and outside of Africa who could help, especially with, say, the DNA sequencing and the TEM analysis. And in fact, in January, I will have an intern starting to work with me full-time who comes from Martha Clokey's lab in the UK. And this will be one of his primary tasks, is to, I don't know if he's on the call or not, I'll have to see, but he will be setting up further surveys and databases to help us understand what needs to be done and who's willing to help, and connecting those, basically network connecting to bring together the right people to characterize these phages so that we could potentially establish a phage bank. And so if you're interested in being part of that networking exercise, you can go to the Phages for Global Health website and sign up for our newsletters. And through those newsletters, we will make people aware of the surveys and the resources that will be coming available. So some of those resources I want to highlight here in an acknowledgements, and then I want to open up for discussion. I will say that most of the people on this slide were involved in a grant submission that we did, trying to get money to initiate a phage bank in East Africa. That grant was not successful, but we will keep looking. And these are people, and this grant was focused for East Africa, where I know from people who have gone through our workshops or others who we've been in communication with, that at least 450 phages have been isolated already by scientists in East Africa. Yay, great job, so exciting. And the people shown here from Kenya, Uganda, Tanzania, and Rwanda have either isolated some of those phages already or indicated their interest in being involved in the phage bank as they continue their phage isolation. But I also want to highlight, there's so many people from other countries who are willing to help, and I'm sure I've missed and not really listed everyone, but these include some of our workshop instructors, some people, Jessica and Jan from Phage Directory, who have been willing to set up phage banks, phage databases, and are actually helping to set up the Australia Phage Bank. There are phage banks, as I said, that already exist in Georgia, Belgium, Canada, Australia, Germany, and Israel, and all of them are willing to help. And I've been talking with them over the past few months about lessons learned. What would they recommend you do or not do in setting up phage banks in Africa? Frank Schmecker at Jafral in Slovenia is also an expert in manufacturing phages and has been more than willing to help advise, and has some specific ideas about how we might be able to do that in Africa. More recently, Balint Kinces at Hungary has also offered to help with a lot of sequencing of phages. So that will be part of our effort starting in January, is to connect a number of people with him to help with some of that. I want to highlight 5 people on this list who I've been really brainstorming with over the last few months. Lillian and Milka at Kemri, together with Michael John Nikolic, who's been working closely with Lillian. Michael John is in the US military, and they have a satellite group that Lillian's a part of in Kenya. And then Martha Clokey, the senior instructor for our workshops in the UK, and also Jessica Nakavuma in Uganda, who hosted our very first workshop and has been helping a number of other groups isolate phages in Uganda. We've all been talking about what's the nitty-gritty of what would it take to actually set up phage banks, starting in Kenya and Uganda. And we welcome input from everyone as we start thinking about that in the new year. I think we can hopefully initiate that in 2022. So stay tuned and let us know if you're interested in being a part of that. I think I'll end with that and take any questions or discussion that we might have. I'm going to stop sharing.
Thank you, Tobi. Thank you very much for the very nice presentation about establishing phage banks. And I think that is the way to go. I know you have tried to make efforts in this area, but I believe in this forum, we can also have some discussions on how to go about it. But generally, because there are other countries that have already established phage bank, we can borrow the best practices from them already so that we don't have to reinvent the wheel. so that it's easy. How did they establish? What do they need? What do we need? So that we borrow the best practices from them. In general, I like the idea. It's good because so that we know which phages we have where, and in case of any need for any patient, we know where to get the phages. I am happy with the already initiatives to establish Phage Bank. I know Dr. Musila, Lilian Musila, whom we collaborate and we work together, has also been very passionate on that. Dr. Professor, you know, now Jessica is a professor, Professor Jessica Nakavuma, who has been my long collaborator for a long time. I'm happy also she's making all the efforts. And I remember she hosted me in her lab for 2 weeks to do phages, for which we did some work, and actually we have already published the paper, is out this week. So yeah, so I want to welcome members of this forum to ask the questions, but maybe before that, I see some 2 questions on the chat. Then we can open. So one question is, this is from Unlib Kiza. Do you think bacteriophages alone has the potential to replace antibiotic use, or is it better to use them in combination with antibiotics? And then the other one is, why don't we have countries from West Africa and North Africa that are also conducting phage research? Maybe Dr. Tobi, you can respond, then we can open the forum for others to ask more questions.
Sure. And let me start with the second question. There definitely are people in West Africa, North Africa, and also in South Africa who are isolating some phages. So I don't want to minimize those efforts as well. We're just aware of most of the work that's been done. And I think the largest number of phages has been isolated thus far by people in East Africa, but we want to help grow the other regions too. And then personally, I think that phages working with antibiotics will be a good way to go. Ben Chan, one of our workshop instructors, has actually shown that you can use phages to push bacteria to evolve to become resensitive to antibiotics. And that's a fabulous thing if we could continue to do that. Ben's shown that now for at least Pseudomonas and Shigella bacteria and phages, and I think that will be applicable to other types of bacteria as well.
Thank you, Toby. Now I will open the forum for further questions. If you have a question, you can just speak.
Well, there's another question I see. Is sequencing a phage enough to characterize a phage? Can sequence data tell which group of phages it is without doing TEM? I think that's a fair question. Generally, it has been considered that TEM should be done to demonstrate that it really is a phage that you're looking at and the characteristics of that phage. Though more and more, I think some scientists are questioning how critical that is. Rob Levine in particular has proposed, from Belgium, has proposed that that may not be as critical.
Yeah, thank you. Any further questions or comments on how we can do this?
I realize we're at time for the next speaker too.
Yeah.
Yes, I just wanted to say good morning, Tobi, and thank you for the time. I think it's important for us to consider seriously the issue of taking a database of phages. I think that idea is very, very important. Let's have a database as mentioned, so that people will know the phages available here and there, so that because I know that as Phage Director we keep sending phage requests for somebody needing phage therapy. We don't have a comprehensive database with regards to Africa. What we have, for example, here in Nigeria, we have a lot of Salmonella typhi phages. We don't know what's available in Kenya, other parts of the world. So I think it's something APF will begin to brainstorm about, to have a central database with regards to phage available in labs, so that it will help in the establishment of the phage bank. Thank you very much once again for your time. And thank you, Dr. Tonga.
Yeah, thank you very much. I also want to thank Ivy, who was supposed to introduce Tobi. And she actually requested me to introduce Tobi because she felt that we have interacted with Tobi more times. So thank you, Ivy. I want to return the ball back to the moderators of today. Thank you, Tobi, and
Sure.
hope to see you in face soon.